Welcome back to the HIV RNA Test Guide podcast. Your trusted source for reliable information on HIV testing, prevention, and treatment.
Absolutely.
And today, we're going to be diving into a topic that's, I think, particularly relevant to our listeners.
So today we're diving into this promising new HIV vaccine candidate. It's being developed by the Duke Human Vaccine Institute, DHVI.
DHVI, right.
Think of it as looking towards a future where maybe the need for testing, or at least the scale of testing, might look different because prevention is stronger.
Okay, so our mission for this deep dive is really to unpack this Duke vaccine. We want to get into how it works, where they are with it, the progress, and what it could actually mean for preventing HIV. We've looked at their innovative strategy, the trial results, the bumps in the road, and the potential global picture.
That's good.
All right, let's jump right in. What's the core idea? What's so innovative about Duke's approach here?
Well, what's really groundbreaking, potentially, is that the Duke team is going after something that's been incredibly difficult to achieve for decades.
Look.
They're aiming to generate what are called broadly neutralizing antibodies. You might see them called BNAbs.
BNAbs. Heard of them? Like super antibodies.
That's a good way to think about it, yeah. They're antibodies that could potentially work against lots of different strains of HIV, the ones circulating all over the world.
Wow. So like a master key, instead of needing a different key for every single lock the virus presents.
Exactly like that. Instead of chasing a virus that's constantly changing its surface, it's disguise.
Which HIV is famous for.
Notoriously so. This approach tries to build a defense that recognizes something more fundamental, something stable.
Okay, that makes intuitive sense. How are they actually trying to do that? What's the mechanism?
They're using a really clever two-part strategy. First, they target specific bits of the virus that are known to be quite stable.
Stable meaning they don't mutate so much.
Precisely. The main target is an area on the virus's outer shell, its envelope protein. It's called the membrane proximal external region, or MPER.
MPER.
Got it. This part doesn't change nearly as rapidly as other parts of the virus's surface, which makes it a much more reliable target, aiming for the Achilles heel, so to speak.
Okay, target this stable spot. What's the second part of the strategy?
The second key piece is something called sequential immunization.
Sequential immunization. Sounds like steps.
It is. Think of it like a very specific training course for your immune system. They give a series of different but related molecules immunogens.
Immunogens. Like harmless mimics of the virus.
Exactly. They look enough like parts of HIV to get the immune system working, but they can't cause infection. They're decoys.
Okay.
And over time, getting these different immunogens in sequence helps guide the B cells, those are the antibody factories in our body, to gradually learn how to make those really powerful, broadly neutralizing antibodies.
So it's not just one shot, it's a process.
Yeah.
Like showing the immune system different clues over time so it finally figures out the master plan.
That's a great analogy. You're guiding it step by step towards producing the right kind of defense.
That does sound really different. And I understand they've had some early signs that this might actually work.
They have, yes. Back in 2019, they ran a Phase I clinical trial. Small group, 20 HIV-negative volunteers.
Phase I, so focused on safety and initial response.
Exactly. And what was exciting was that they actually saw the very beginning stages of BNAb production starting in some participants, and this was within weeks.
Within weeks?
Yeah. It was a crucial first step, kind of a proof of concept that their strategy could kickstart this process in people.
Okay, that sounds genuinely promising. A proof of concept. So, building on that, where are things now with testing? What happened next?
Well, the initial trial did hit a snag. They had to pause it.
Oh, why?
There was a non-life-threatening allergic reaction in one of the participants.
Okay, safety first, always.
Absolutely. And the investigation suggested it was likely caused by an additive in the vaccine mixture, not the core immunogen itself.
Right, so something in the supporting cast, not the main actor.
Seems that way. So what they're doing now is obviously reformulating. They're working on refining the ingredients to make sure that doesn't happen again.
Makes sense. And what's the plan going forward? Just tweak that one.
The bigger plan, looking ahead, is actually to develop what they're calling a multi-component vaccine regimen.
Multi-component, so more than one target.
Exactly. The idea is to have a vaccine with several different parts, maybe each targeting a different vulnerable spot on the virus, like the MPER and potentially others.
Like a combination attack.
Precisely. The thinking is that hitting the virus from multiple angles at once could make the vaccine much more potent and harder for the virus to escape.
That sounds like a more robust strategy, for sure. What kind of time frame are we talking about for seeing these refined versions in trials?
Well, according to Dr. Mihai Azoite at DHVI, they're hoping human trials for these improved candidates could potentially start in the next, say, 3 to 5 years.
3 to 5 years for the next round of human trials. Okay.
And then, if things go well, potential public availability. Well, that's likely further out, maybe 5 to 10 years from now. So pushing into the early 2030s, maybe around 2032.
Okay, 2032 ballpark. So definitely not tomorrow. But it feels like tangible progress is being made.
It does feel that way, yes.
Now, you mentioned this is different from earlier attempts. Why has an HIV vaccine been so incredibly hard to develop? What made those previous efforts struggle?
Oh, it's been a massive challenge. There are a couple of huge reasons. First, as we touched on, HIV mutates incredibly fast.
The constantly changing target problem.
Exactly. It's like trying to shoot down a target that changes shape and color every second. Traditional vaccine approaches often couldn't keep up.
Right.
And second, HIV is a retrovirus. This means it actually inserts its own genetic code right into the DNA of the human cells. It infects our immune cells, ironically.
It hides inside our own cellular machinery.
It does. It becomes part of the host cell, making it extremely difficult for the immune system to find and eliminate all infected cells.
So a constantly changing enemy that also hides inside your own defenses. Yeah, I can see why that's tough.
It's a formidable combination. So previous vaccine attempts often struggled to generate an immune response that was broad enough to cover all the variants and strong enough to overcome this hiding strategy.
Which brings us back to Duke's approach. How does it sidestep those specific problems?
Well, by focusing on those stable regions like MPER, they're trying to bypass the rapid mutation problems some way. That target doesn't change as quickly, and the sequential immunization. That's the attempt to train the immune system more effectively, to coax it into making those powerful BNAbs that might be better equipped to neutralize the virus despite its defenses. Using those synthetic mimics is also key. It's a more directed way to teach the immune system.
It really does sound like a different playbook, learning from decades of research. But even with this promising strategy, it's not a done deal. What are the big hurdles they still need to clear?
Oh, definitely several major challenges remain. A big one is boosting the antibody levels.
Boosting the BNAb.
Yeah. That phase I trial showed they could be initiated, which was huge. But for real-world protection, you need much higher levels of these BNAbs, and they need to be potent, really effective at neutralizing the virus.
Right, you need enough soldiers, and they need to be strong soldiers.
Exactly. So enhancing the quantity and quality of the BNAb response is critical.
Okay, what else?
Safety, of course, remains top priority. They need to ensure the refined formulations, especially the multi-component ones, are safe and well-tolerated in larger groups of people. That allergic reaction highlighted the need for careful formulation.
Careful checks.
Then there's the sheer complexity. Designing, manufacturing, and delivering a vaccine with multiple distinct components working together. That's scientifically and logistically much harder than a single component vaccine.
Yeah, I can imagine coordinating all those parts is a challenge in itself.
It adds layers of difficulty.
And then there's the whole process of getting it approved.
Absolutely. Regulatory approval is a massive hurdle. You need large-scale, rigorous clinical trials, phase 2 and especially phase 3, to prove definitively that the vaccine is both safe and effective at actually preventing HIV infection in the real world.
Thousands of people, long-term follow-up.
Yes, it's a very time-consuming, very expensive process. Regulatory bodies like the FDA need rock-solid evidence before they'll approve a vaccine for public use.
So, quite a journey still ahead, even with the exciting science. Let's zoom out again. Thinking about our listeners, many connected with the HIV RNA Test Guide podcast, involved daily in testing and helping people know their status. How could an effective vaccine like this eventually change the global picture of HIV prevention? How does it fit with testing?
Well, if this pans out, if we get a truly effective HIV vaccine, it would be monumental for global health, just transformative.
How so?
Its main potential is obviously to dramatically cut down the number of new HIV infections happening worldwide.
That's the ultimate goal of prevention.
It is. And it wouldn't make existing strategies obsolete, not at all. Things like pre-AP, condoms, harm reduction, treatment as prevention, they'd still be vital. But a vaccine would add another incredibly powerful layer of defense.
Like adding a shield to the existing armor.
A very strong shield, hopefully. Think about reaching those UNAIDS 95 goals, diagnosing 95% of people with HIV, getting 95% of them on treatment, and 95% of those achieving viral suppression. A vaccine preventing new infections would be a massive boost towards those targets.
Yeah, it tackles the front end of the problem.
Exactly. And beyond the direct health impact, consider the economic and social benefits. Reducing the number of people needing lifelong HIV treatment saves huge healthcare costs. And reducing infections could, over time, help significantly reduce the stigma that still, sadly, surrounds HIV.
And for the testing community.
Testing will always be crucial for public health, for diagnosing existing infections, monitoring treatment, identifying other STIs. But if a vaccine dramatically lowers the number of new infections, the incidence rate,
Then the focus or scale of testing might shift over the very long term.
it could potentially evolve, yes. Maybe less focus on widespread screening in some populations if transmission rates plummet, perhaps more focus on specific groups or diagnostic testing. But that's looking quite far ahead. Testing remains fundamental now and for the foreseeable future.
A really important perspective. It could reshape the landscape, but complements current efforts. So what are the immediate next steps for the Duke team? What are they working on right now?
As we touched on, the main focus is developing and refining that multi-component vaccine idea.
Getting the formula right for that combination attack.
Yes. They're designing and testing different combinations of immunogens, potentially targeting 3 or even more distinct stable sites on the virus. The goal is to create a regimen that trains the immune system to produce much stronger, much broader neutralizing antibodies than they saw initially.
Really building on that proof of concept to get a more powerful response.
That's the aim.
Okay, let's quickly revisit the timeline. We heard the 3-5 years for next trials, maybe 5-10 for availability. Can you just briefly walk us through the standard vaccine trial phases again?
Sure. It's a structured process. Phase 1, like Duke completed, is small, maybe a few dozen people. Focus is safety, dosage finding, getting early signals of immune response. Phase 2 is larger, hundreds of people. It looks more closely at safety across a wider group and gets better data on the immune response. Does it generate the antibodies we want? Phase 3 is the big one. Thousands, sometimes tens of thousands of people, often across different countries. This is where you really find out. Does the vaccine actually prevent infection compared to a placebo? And you watch very closely for any rarer side effects.
That's the make or break phase for efficacy.
Absolutely. And then, even after approval, there's often phase 4, which is post-market surveillance, monitoring safety and effectiveness as it's used in the real world in millions of people.
Right. So Duke finished phase 1 with the initial concept.
Yes. And now they're essentially back in the development stage, refining the product before launching into phase 2, and then hopefully phase 3 with the new multi-component version.
Which they hope to start in 3-5 years.
That's the current projection. And if those large phase 3 trials are successful, then you're looking at regulatory review, which takes time, leading to that potential approval maybe 7 to 10 years from now, around 2032.
Is there any chance of speeding that up?
Sometimes, if results are exceptionally strong for a condition with a high unmet need, regulatory agencies might offer accelerated pathways. But you still need robust data. The 5-10 year time frame is probably the most realistic baseline to think about.
Okay, so bottom line. Exciting science, real progress, but public availability is realistically still a number of years away.
That sums it up well, yes.
So let's wrap up by bringing it back to the potential impact one last time. If this, or something like it, gets approved, summarize the revolution.
Okay, the big wins. First, huge potential to reduce new HIV infections globally. We're talking about a virus affecting nearly 40 million people right now. That's massive.
Life-changing potential.
Absolutely. Second, it powerfully boosts our existing prevention toolkit, PrEP, condoms, treatment as prevention, adding another strong layer. Third, it helps us get closer to those global health goals, like the UMAIDS targets. A major step forward.
And fourth,
significant economic and social relief, lessening the financial burden of lifelong treatment, and, crucially, chipping away at the stigma associated with HIV.
a truly hopeful outlook.
Yeah.
Okay, so bringing this deep dive to a close, we've really dug into Duke's innovative strategy for an HIV vaccine, those promising, though early, results.
And the challenges, too.
And the challenges? Yes. The allergic reaction, the need for stronger responses, the regulatory hurdles, and finally, the potentially massive impact this could have down the line.
It highlights the incredible science happening.
It really does.
Yeah.
And it's a reminder that while everything we do now, testing, treatment, existing prevention, the crucial work many of our listeners were involved in, remains absolutely vital.
Essential.
This kind of research offers real, tangible hope for changing the future trajectory of the HIV epidemic.
Couldn't agree more. It underlines the dedication out there. But until these breakthroughs arrive, sticking to what we know works, testing, treatment, prevention, is paramount.
Well said. And maybe a final thought for everyone listening, given this potential timeline, 5, 10 years out, and how HIV prevention is always evolving, what questions does this raise for you about the future? How do you see a tool like a vaccine fitting alongside testing and current strategies in, say, 2035? Definitely something worth pondering. Thank you for joining us on this deep dive.